This conference will now be recorded.
Yes, I'm audible. Welcome to
yes,
all the
ma'am.
participants. In last class, students, we studied about the hormones. Mostly students, I think some students were in the class. First of all, we must know what are the hormones. In last class, we studied and we discussed about the hormones. Okay. First of all, everyone must know what is a hormone. Hormone is the growth regulator. Clear? In plants as well as in animals, the hormones are the growth regulators and they are regulating different activities. Okay? In plants as well as in animals, in our body, there are lots of hormones are present. Okay, like growth hormones, insulin hormone, glucagon hormone, and many more hormones are present in our body also. In the same way, in the plants, many regulators are present, and these regulators which are regulating the growth are called the hormones. In other words, the hormones are also called as the plant growth regulators, as well as they are phytohormones. Phytohormones as well as plant hormones, both of these are the same term. Don't get confused. Phytohormones as well as plant growth regulators and hormones, these are the same terms. Firstly, what are the hormones? Hormones are the growth regulators. They are organic compounds. They may be naturally synthesized or they may be artificially synthesized. Okay? उनको plants naturally भी synthesize कर सकते हैं और they may be synthesized artificially. Okay, and they are mainly used to control the different activities and many physiological process in a plant. Okay, hormones are responsible for the growth as well as for the different physiological activities, from the seed germination to the flowering to the abscission of leaf. Abscission of leaf means the falling of leaves. आप देखते हैं कि leaves fall करते हैं plant से. All of these processes are mainly occurs in the presence of hormones. Okay, so students, keep in mind that hormones are the most important compounds which are regulating the most of the activities. Hormones are regulating the almost many activities in the plants, from the seed germination to the fruiting, flowering and abscission of leaves. All of these activities are regulated by the plant growth regulators. Growth regulators doesn't mean that they are only regulating the growth. They are regulating almost all the processes in the plants. I hope definition is clear to all the students. Now, they are also called as phytohormones, as I already told you that hormones are also known as phytohormones because they are present in the plants and they are messengers. They act as the physiochemical messengers which regulate the germination, rooting, growth, flowering, fruit ripening, senescence, death, seed dormancy. All of these processes are regulated by the hormones. Then hormones have different features and different characteristics are present in the hormones. Okay, the hormones have different features and many other characters are present in the hormones. What are the different features? First of all, the hormones are required in very low amount. Okay, ऐसा नहीं होता कि hormones की बहुत ज्यादा requirement है. In seed germination the requirement is low and in the fruit arrival hormone requirement is high. It doesn't happen. The concentration of hormone which is required for the plant growth is always very low. It vary from 10 to power minus 6 to 10 to power 5 molar. Okay, the hormones concentration is very low and the requirement is very less as compared to the requirement of the minerals and vitamins for the growth of the plant. Okay, like in for the growth of the plants we require different types of minerals and vitamins. We are providing different minerals to the plants. Okay, we are giving water to the plants. All these are required in high concentration, but on the other hand the hormones are required in very low concentration. Okay, the hormones are maybe synthesized naturally in the some plants of the part. ओके, अब हमारे माइंड में क्वेश्चन अराइज होता है, हॉर्मोन्स हम आर्टिफिशियली भी देते हैं प्लांट्स को और वो उसके अंदर ही सिंथेसाइज होते हैं. बोथ ऑफ दीज आर द क्वेश्चंस व्हिच अराइज इन अवर माइंड when we are doing the practicals and research experiments. At that time, the treatment of hormone is also given from the outside. Maybe artificial treatment of the hormone is given to the plant. Suppose if we want to check the growth of the plant, then we are providing the hormone, like gibberellin hormone is given from the outside. From outsources we are giving the gibberellin hormone. On the other hand, hormones are also synthesized naturally in the plants. It depends on the requirement of the plants, on the process they are regulating. Okay, it depends on the processes which are regulating. If leaf abscission is the process, suppose at some time the abscission of leaf takes place. हम कभी भी जब आपने देखा प्लांट से लीफ गिरते हैं, we are not giving any hormone, abscisic acid, we are not giving the hormone, but at that time the plants are naturally synthesizing these hormones. So plants may be provided artificially or they can be synthesized naturally in their own body. Okay, and the hormones are more diffuse and they are not localized. Hormones are more dynamic in nature, they are not static. Static नहीं होते hormones, they are always dynamic in nature. जहाँ पे इनकी requirement होती है, the site where the hormones are required, they reach at the site and perform their function. Okay, hormones are always dynamic and they are more diffused. The hormones are always more diffused. Clear? They are present in different parts from the root to the leaf. They can travel at any place because their amount is very low, and when at the specific location where they are required, they reach at the site and perform their function. So the hormones are much more diffused. Then the next point, different types of hormones. Hormones are of different types: auxin, gibberellin, cytokinin, ethylene, and abscisic acid. These are the main 5 hormones which we study in case of plants. In addition to that, brassinosteroids, jasmonic acid, all of these are also acts as the plant growth regulators, and we also call them as the plant regulators, phytohormones. Okay, in addition to these main hormones, auxin, gibberellin, cytokinin, ethylene, as well as abscisic acid. These are the main hormones, the 5 main hormones we always studied. Okay, and these hormones perform different functions. Let's take the overview here. Suppose first hormone is auxin. In last class we discussed about the auxin hormone. Okay, some of the functions we discussed, we will later on discuss the other functions as well as their biosynthesis pathway. Okay, we later on discussed in next class, maybe we discussed about the auxin hormone, the biosynthesis pathway. Okay, next is gibberellin. Gibberellin hormone, the main function is it helps in the elongation, it helps in the growth of the plant. Cytokinin, in addition to this, there are many more functions of the hormones, but these are the main functions. Like cytokinin, this hormone mainly helps in the process of cell division as well as in the cell growth. Okay, cytokinin hormone, it mainly helps in the process of cell division as well as in the cell growth, cell senescence. These processes are regulated by the cytokinin hormone. The next hormone is ethylene hormone. The main feature of ethylene hormone is it is gaseous in nature. Okay, it is gaseous in nature. It is the only one hormone which is gaseous in nature, and it mainly helps in the process of fruit ripening. Then next is abscisic acid, ABA hormone. This is the stress hormone. ABA hormone is the stress hormone. Okay, clear? These are the main 5 hormones we studied: auxin, gibberellin, cytokinin, ethylene, and abscisic acid. In addition to this, there are many other hormones like jasmonic acid, brassinosteroids, and some hormones are also present which also regulate the growth of the plant and regulate the activities of the plant. Okay, then we classify these hormones. Okay, we are classifying these hormones because all the hormones are not promoters. Out of these hormones, some acts as inhibitors and some acts as the promoters. Okay, so On the basis of their promoting activities and inhibiting activities, they are classified into the two categories, promoters as well as inhibitors. Promoters mainly help to growth of the plant. Promoters, name indicates itself. Inhibitors, they are mainly used to reduce the growth, and different functions are reduced by the inhibitors. Okay, promoters and inhibitors, these are the two categories in which we classify the plant hormones. ऐसे नहीं होता बच्चों की hormones होते हैं तो mainly growth regulators है, तो mainly they are regulating the growth and they are promoting the growth and promoting the different functions. They may also act as the inhibitors. अगर hormones activators का काम करते तो plants में कोई मतलब negative activity नहीं होती, leaf fall नहीं करते, and all these activities will never take place. To control the promoters, inhibitors also work. Okay, ऐसे होता है बच्चों की जो promoters होते हैं उनकी एक्टिविटीज को रेगुलेट करने के लिए भी also require the inhibitors. So hormones act classified into the two categories, promoters as well as inhibitors. Promoters which increase the growth and they help to grow the plant faster. And promoters are auxin, cytokinin, gibberellin, the steroids. All of these are the promoters. Inhibitors which reduce the growth. Inhibitors are ethylene, abscisic acid and jasmonic acid. These are the inhibitors. So today we are going to discuss the one hormone, that is gibberellin hormone, the basics and biosynthesis. First of all, we must focus on the biosynthetic pathway, because from the biosynthetic pathway all the time many questions are arising. Okay, the pathway is malonic acid pathway and different pathways, and out of these pathways at every year, at every session, lots of questions are arising from this the biosynthetic pathway. So today we are going to discuss the one growth promoter, that is gibberellin hormone. शॉर्ट फॉर्म इज GA. यहां तक सबको क्लियर है? किसी स्टूडेंट को डाउट है तो कमेंट करें. कोई डाउट है यहां तक? डेफिनेशन हॉर्मोन्स का बेसिक कंसेप्ट इज क्लियर टू ऑल द participants, to all the students? Up to here, if any one of you have any doubt, then you are free to ask. Up to here, clear to all the participants? Yes, Shalini, clear? Other students? These are the basic concepts. Yes, okay, I hope clear to all the, okay. So these are the basic concepts, students. What are hormones? We discuss their definition, then their different types and their quantity, in how much amount they are required. Okay, then our first hormone, that is gibberellin. All of you are aware with the name gibberellins. सबने नाम सुना होगा gibberellins. Gibberellin hormone, they are the plant growth regulators and they regulate different processes, like it help in the stem elongation, germination of the seed, it brings the dormancy of the seed, flowering, sex expression. How it helps in the sex expression? Because देखो बच्चों, hormones को different concentration में देते हैं. ऑक्सिन एंड जिबरेलिन रेशियो होती है. अगर उस रेशियो में ऑक्सिन ज्यादा है तो दूसरा maybe male develop हो. When concentration of gibberellin is higher, maybe development of the female flower takes place. So it helps in the sex expression. It helps in the induction of the enzymes, leaf and fruit senescence. All of these are the tasks which are performed by the gibberellin hormone. So gibberellin are the plant growth regulators which regulate the developmental processes. All the developmental processes are regulated with the help of the gibberellin hormone. Okay. The basic functions are regulated by the gibberellin hormone. Then the next question that comes in our mind, gibberellin hormone, they are the diterpene plant growth hormones. Diterpene. The main point that they are the diterpene and they are active due to the presence of this COOH group. This COOH group mainly helps in the activation of the gibberellin hormone. This carboxyl group is mainly responsible for the activity of the gibberellin hormone. Okay, students, यहाँ पे हम एक point, we are using the pen. Gibberellin hormone are of two categories, 19 carbon and second is 20 carbon. 19 carbon as well as the 20 carbon. Gibberellin are classified into the two classes which contains a 19 carbon as well as a 20 carbon. Out of these, the 20 carbon form is the most active and biological active form of the gibberellin. Okay. Gibberellin is a diterpene hormone and it is the most important hormone. And one point keep in mind, students, you can also note these points in your notebook. These are the largest known class of plant hormones. Gibberellin are the largest known class of hormones. Out of the other hormones, we have studied the plant promoters as well as inhibitors. Out of these, the gibberellin is the most diverse, most largest group of hormone. Largest class of hormone is gibberellin. Okay, and mainly two forms are studied: 19 carbon and 20 carbon. Two classes of hormones are gibberellin hormone is present. One is 19 carbon and other one is 20 carbon. Out of these two forms, the 20 carbon form is most active form. Okay, the 20 carbon form is the active form, and the carboxyl group at the 7th position, first, second, 3, 4, 5, 6, 7. At the 7th position, carboxyl group which is present is mainly responsible for the activity of the gibberellin hormone. So बच्चे, मैंने अभी आपको बोला the gibberellin hormone is the most largest known hormone. It is the largest known hormone, largest family of the hormone. So ऐसा तो नहीं है कि इसकी सभी forms active होंगी. The form which contain the carboxyl group is the active form. Okay, so gibberellin are the diterpene compounds and they are plant regulators which regulate the growth and development, and it is the largest class of hormone. It is classified into the two classes, 19 carbon and 20 carbon. Out of these two forms, the 20 carbon form is the most active form. The 20 carbon form is the most active form, and the carboxyl group is mainly responsible for the activity of this hormone. Carboxyl group which is present in the 7th position is mainly responsible for the activity of the hormone, and this carboxyl group is mainly responsible to make the hormone active. Okay, next point. Then the next point arises in our mind, the production, the site of production, at which site the hormone brain is produced. As we studied in case of auxin, the auxin is synthesized in the stem part and its movement is always unidirectional. Okay, so the gibberellin hormone, it is always synthesized in the young tissue, germinating seeds, developing shoot tissue, embryo, and its production increase in the dark. Okay, students. So its production is mainly at the young growing tissues, maybe root, seeds, the seeds which are germinating. It is the one hormone which helps in the germination of seeds. Seeds are germinating. First of all, they break the seed coat. After that, the seedling is coming out from the seed. So this hormone, this gibberellin hormone, mainly helps in the germination of seed. So the production of gibberellin takes place at the young parts, the growing parts, like young tissue of the roots. The young tissue of roots which are producing is produced at that site. At the germinating seeds, developing shoot tissues, at the embryo. And its production always increases in the dark. As in the auxin hormone, it was studied that auxin की discovery ही कैसे हुई थी? The Darwin scientist who discovered the auxin hormone. The auxin hormone, the bending of the plant part is towards the light. That bending mainly arises at discovery of the auxin hormones because towards the light, bending of the plant part towards the light is mainly due to the auxin hormone. In the same way, the gibberellin hormone are always produced in the dark part. Okay, they are always produced in the dark. And their acetyl CoA, we will discuss this point later on. This is the important point from acetyl CoA. I will clear this point. First of all, you must know that the gibberellins are the tetracyclic and diterpenoid. Please note these points in your notebook. The gibberellins are tetracyclic. Clear? These are the tetracyclic. 3 cyclic structures are present. tetracyclic. 1, 2, 3, and this is the basic methyl group present. So it is a tetracyclic diterpenoid. It is tetracyclic diterpenoid acid which is synthesized by the terpenoid pathway. Its synthesis occurs with the help of the terpenoid pathway. Okay, keep in mind that the synthesis of gibberellin occurs from the terpenoid pathway, and it is synthesized in the plastids as well as in the endoplasmic reticulum and cytosol, and after reaching to the cytosol they get activated. I will explain the pathway, bachcho. Firstly, keep in mind that there are the 3 sites for the production of gibberellin hormone. Mainly they are produced at the growing tissue, the young tissues, young parts of the roots, the germinating seeds, the shoot tissues, the embryo, and the location in plants. What is the location? They are mainly synthesized. Synthesis कहाँ होता है उनका? In plastids, in endoplasmic reticulum, as well as in the cytosol. And after reaching to the cytosol, they get activated in plastid and endoplasmic reticulum. They are synthesized. Their different molecules are synthesized, but after reaching to the cytosol, they get biologically active. साइटोसॉल में जाने के बाद ही बायोलॉजिकली एक्टिव होते हैं स्टूडेंट्स. सो फर्स्टली कीप इन माइंड दीज आर द साइट्स. ये सबको क्लियर हो गया. मेनली इट इज प्रोड्यूस्ड एट द यंग ग्रोइंग साइट्स, द यंग टिश्यूज, द जर्मिनेटिंग सीड विच इज जर्मिनेटिंग इटसेल्फ, द डेवलपिंग शूट टिश्यू विच इज डेवलपिंग. ऐसे नहीं कि डेवलप्ड पार्ट्स में कभी नहीं प्रोड्यूस होगा जिबरेलिन बच्चों. Keep in mind that gibberellin is always produced in the developing parts, developing root, germinating seeds, not germinated seed. Germinating seed. Keep in mind the seeds which are germinating itself. Developed shoot tissue nahi bachcho, developing shoot tissue. So the gibberellin hormone is always produced in the growing parts. Okay. Then this is the main point. I was discussing this point. Gibberellins are diterpenoid. And they are tetracyclic, diterpenoid as well as they are tetracyclic. They are mainly synthesized by the terpenoid pathway. Okay, they are mainly synthesized by the terpenoid pathway in the plastids. Their synthesis starts in the plastid, modified in the endoplasmic reticulum, and in the cytosol they become biologically active. Clear, students? Just minutes, I'm comment me in message box. किसी को यहाँ तक कोई डाउट है? Any doubt to any one of the student? आपकी कमेंट्स लेना भी जरूरी है. एनी नो डाउट एनीवन? ओके. So gibberellins are the diterpenoid and tetracyclic compounds. किसी स्टूडेंट ने कुछ क्वेश्चन पूछा है? जस्ट अ मिनट, वन क्वेश्चन. Diterpenoid, these are the compounds which are heterogeneous group of compounds. Diterpenoids are the heterogeneous group of compounds which contain the different number of carbon atoms. So they may be bicyclic, they may be tricyclic, tetracyclic, pentacyclic, hepta. The diterpenoids are the compounds. Okay, so gibberellins are the diterpenoid acids and they are mainly synthesized by the terpenoid pathway. The pathway which helps in the synthesis of gibberellin acid is terpenoid pathway. Okay, बच्चों ये क्वेश्चन पूछते हैं. The site of the synthesis of the gibberellin hormone, plastids, ER, cytosol. Cytosol, plastids, ER. Confusion will be there. So keep in mind that first of all they are synthesized in the plastids. Then they are modified in the endoplasmic reticulum. Then after modification in the endoplasmic reticulum, they are moved in the cytosol. In the cytosol, they become biologically active form. In the cytosol, the inactive forms are always converted into the biological active form. So gibberellic acid is dihydroxylated gibberellin. Just a minute. Dihydroxylated means they contains 2 hydroxyl group. Di means 2, hydroxyl group I think सबको पता है, OH group. They are dihydroxylated group. One hydroxyl group is present and other hydroxyl group is present here. So gibberellic acid is dihydroxylated gibberellin, and this hydroxylation also affects the activity of the gibberellin. Okay, these hydroxyl groups are also affect the activity of the gibberellin hormone, and these points you keep in mind that the gibberellic acid which is present, the gibberellic acid at which the hydroxyl group is present at 3rd and 13th position is the most active, that is gibberellin 3. Students, this is the most active gibberellic acid. Okay, I told you that gibberellin is the largest hormone. It is the largest class of hormone. सबसे लार्जेस्ट क्लास है जिबरेलिन एसिड. तो ऐसा तो नहीं होता बच्चों, इसमें जितने भी फॉर्म है ऑल द फॉर्म्स आर एक्टिव. ओके, सभी फॉर्म्स तो इसके एक्टिव नहीं हो सकती, कुछ फॉर्म इनएक्टिव होती हैं, कुछ फॉर्म लीस्ट एक्टिव होती हैं, सम फॉर्म्स आर मोस्ट एक्टिव. सो हाउ कैन वी क्लासिफाई द एक्टिविटी, हाउ कैन वी क्लासिफाई द एक्टिव फॉर्म? The active forms are classified on the basis of the hydroxyl group, the position of the hydroxyl group, and the most active gibberellin hormone is which contain the dihydroxyl group, one hydroxyl group and second hydroxyl group. This is the dihydroxyl gibberellin, and the most active gibberellin is which contain the hydroxyl group at the 3rd and 13th position. This is the most active gibberellin form, that is gibberellin 3. Out of all the gibberellin, out of, if there are 200 forms of gibberellic acid are present, out of all these forms, the gibberellin 3, GA3 is the most active form. And why it is most active? Because it contains hydroxyl group at the 3rd position as well as on the 13th position. And this dihydroxylated form make it the most active compound, most active gibberellin. Out of all the gibberellins, the gibberellin 3 is most active. Clear? And on the other hand, ethylene hormone, the ethylene hormone, that is the gaseous hormone. Note this point in your copy. Students, all the points are not given in the slides. Some of the points जब हम पढ़ाते हैं तो वो माइंड में आते हैं from the previous year questions, from the previous knowledge. We are remembering the points. So these points which are not mentioned in the slide, you note in your notebook, write down your notebook that ethylene hormone decrease the concentration of bioactive gibberellin. The hormone acts as the inhibitor of gibberellin. So ethylene hormone is the inhibitor of the gibberellic acid. Bioactive gibberellin hormone inhibited by the ethylene. Students, note this point. Okay, I'm writing the point here. Ethylene. Ethylene. This hormone decreases the concentration of bioactive gibberellin acid. Students, clear? These points are clear to all the students? अभी बच्चों हम बेसिक देख रहे हैं इसका. बेसिक, फर्स्टली वी क्लियर द बेसिक. Basics are also most important. Okay, so gibberellin 3 is most active. Why it is active? Because the presence of the dihydroxyl groups. 2 hydroxyl groups are present, which makes it the most active form. Production we have studied. Then these are the points. They are synthesized in the plastids, modified in the endoplasmic reticulum, and they get activated in the cytosol. The gibberellins are derived from the gibberellin skeleton, but synthesized by the and curin. ये बच्चों मैं आपको क्लियर करूंगी, तब आपको क्लियर होगा. Synthesis and metabolism. They are synthesized by the 2 pathways: mevalonic acid pathway in cytosol and non-mevalonic acid pathway in plastid. Okay. Their synthesis takes place in 2 locations, cytosol and plastid. In the cytosol, they are synthesized by the mevalonic acid pathway, but in case of plastids, they are synthesized by the non-mevalonic acid pathway. Just a minute, I forget the discovery. Students, discovery ki slide, I think slide get deleted. First of all, we are studying the gibberellin hormone. The next question that comes in our mind, how the gibberellin hormone is discovered. Gibberellin hormone को discover किसने किया, discover कैसे हुआ था? It is very interesting story. The slide of discovery is displaced from here. So just discuss the discovery. The discovery of the hormone is most important and very interesting. It was discovered in Japan by the Kurosawa. Kurosawa was a scientist who was working in Japan. He studied the fungal disease called the foolish seedlings disease. Okay, the one of the fungus that affect on the rice plant, and the rice plant are increasing in height. Okay, they are increasing in height foolishly, so it is called the foolish seedling. वो पागलों की तरह अपनी हाइट उनकी बढ़ती जा रही थी. Okay, and the name of fungi was Fusarium fusicora. Okay, it is also called as the Bacchane disease. This disease was also asked the Bacchane disease. Bacchane disease is the fully shielding disease in which the growth of the plant is increasing. There is drastic increase in the height of the plant. Okay, then the Kurosawa scientist, when he was working on in Japan, he was seeing that on the rice plant there is a fungal infection. The fungus was the Fusarium fusicori. This was the fungus, and due to the effect of this fungus, due to the infection of this fungus, the plant height was increasing. The plant height was increasing and increasing, and this disease was called as the Bacchane disease, or it is also called as the foolish seedling disease. Why it is foolish seedling disease? Because the plant was growing madly. ऐसे वो increase होते ही जा रहे थे. So from the fungi, the compound was isolated, the called Gibberellia fusicori. Okay, from the fungi, the compound which is mainly responsible to increase the growth height of the plant was Gibberellia fusicori. The fusicori substance is mainly fusicori. क्यों बोला बच्चों? क्योंकि the fungus name was the Fusarium fusicori. So the compound which was isolated from the fungi was Gibberella fusicori. Later on, it was discovered that the fungi is releasing the substance which is mainly responsible for the growth of the plant. And from this fungi, the Kurosawa scientist discovered the gibberellin hormone. So the first source of the gibberellin hormone was the fungus. जो first source था, that was the fungus. Clear, students? Discovery समझ में आई सबको? Let me repeat. The scientist Kurosawa, working in the Japan, in the rice field, he studied the some of the fungi. One fungi called Fusiferium fusicori is infecting the rice plant, and due to the infection, plants की height बच्चों बहुत ज्यादा बढ़ गई थी, and the disease was called Bacchane disease. Excess of everything is dangerous, na? Height was increasing very rapidly, and drastic increase in the height takes place. So the scientist isolated the compound from the fungi, and he found that Gibberella fusicori. On the name the fungus, the name was given Gibberella fusicori. This is the compound which is synthesized from the fungus. So the first discovery source was the fungus. The first gibberellin was isolated from the fungi, and till currently 136 gibberellins are identified from the plants, fungi and bacteria. पहले एक ही उसको डिस्कवर किया था, but currently almost 136 compounds of gibberellin are synthesized and they are identified. And out of these forms, 136, GA3, it is the most active form. Out of all these 136 form, the gibberellin 3 is the most active form. Okay, then comes the synthesis. The synthesis of gibberellin. The precursor is IPP, that is isopentyl pyrophosphate. Just a minute. Wait, I'm sharing the image with you. One image, I'm sharing the image. I take the screenshot and it is on my email ID. Wait for 2 minutes, students. If anyone of you Kurosawa, yes, Drashta Kurosawa. On my Gmail, I take the screenshot from one of the site in which the pathway is clearly mentioned. The biosynthesis pathway is clearly mentioned on that. Okay, students. So note that the precursor is acetyl-CoA. The acetyl-CoA last slide बच्चों हमने देखा था last slide from the very first slide. यहाँ पे आपको दिखा था synthesis is acetyl-CoA. मैंने आपको बोला था कि मैं I will discuss the point later on. Yes, synthesis is from acetyl-CoA. We discussed this point. The synthesis is from the acetyl-CoA. Okay. Just a minute. I just want to clear the synthesis because synthesis is the most important. I am take that screenshot from somewhere and that's why I am clarifying the point here. I was not able to fit the screenshot in the presentation. That's why the problem is because I was trying to fit the image in the presentation, but the size of the image was very large. That's why I was not able to fit the image here. Yes, I think screen is visible to all the participants. So this is the full psych pathway. We are going to discuss the pathway step by step. We are going to discuss the pathway step by step. मैंने बहुत सारी साइट से देखा तो आफ्टर डिस्कवरी ये जो स्लाइड थी दिस इमेज इज द बेस्ट. इसमें इसका सिंथेसिस पाथवे है वो बेस्ट एक्सप्लेन. सिंथेसिस पाथवे इज बेस्टली एक्सप्लेन एट दिस पॉइंट. फर्स्टली द प्रिकर्सर इज IPP, isopentyl paraphosphate, that is a 5 carbon compound. Okay, the biosynthesis occurs in the 3 stages: plastids, endoplasmic reticulum and cytosol. Okay, plastid, endoplasmic reticulum and cytosol. The biosynthesis includes 3 stages in different organelles: plastid, ER and cytosol. So firstly we start from the very first point, that is IPP. Isopentyl paraphosphate is the precursor, which is a 5 carbon compound. Okay, from this isopentyl paraphosphate the next compound that is synthesized is dimethyl allyl diphosphate. Then one paraphosphate IPP compound is added here. Then the formation of geranyl diphosphate takes place. These questions are almost all the time. One question is asked from here. The 10 carbon compound is geranyl diphosphate, GPP. From the IPP, the formation of GPP takes place. From this GPP, IPP add then formation of pherencyl pyrophosphate because it is a 10 carbon compound. Then one 5 carbon compound is added here. Then the formation of pherencyl diphosphate takes place. This pherencyl diphosphate is 15 carbon compound. From this pherencyl diphosphate, the formation of geranyl diphosphate takes place, which is a 20 carbon compound. This 20 carbon compound is converted into the ant-chlorine, and this ant-chlorine is transported into the endoplasmic reticulum and it is modified here. And gibberellin 12, it get formed here. And in the cytosol, the hydroxylation process takes place. In the cytosol, the hydroxylation process takes place. The hydroxylation of carbon 13 takes place and it forms the bioactive gibberellin. Okay, the formation of active gibberellin takes place in the cytosol. So students, this pathway is most important pathway. Firstly, IPP, isopentyl pyrophosphate. This is the precursor. The first compound is IPP. It is a precursor which is a 5 carbon compound. From this 5 carbon compound, dimethyl allyl diphosphate is formed. Okay, the diallyl diphosphate is synthesized. From here the process is start. First of all, the IPP, isopentyl pyrophosphate, is added. Then the formation of 10 carbon compound it takes place, that is geranyl diphosphate. In the geranyl diphosphate, the other 5 carbon compound is added. Then pherencyl diphosphate is formed. Then geranyl diphosphate. From this geranyl diphosphate, the ant-chlorine is formed. It is transported and it is oxidized. In the plastid, then in the endoplasmic reticulum, it is transported and oxidized. and from the endoplasmic reticulum it is moved in the cytosol, and in the cytosol the process of hydroxylation takes place. So in the cytosol, बच्चों मैंने आपको बताया था, as I told you, the hydroxylation process makes the gibberellin acid active. So in the cytosol the hydroxylation process takes place, and due to this hydroxylation process the gibberellin acid get active. Okay, the gibberellic acid is active in the cytosol. So keep this pathway in your mind: zerenyl pharancyl, zerenyl diphosphate, and curine. Let's move on the presentation. In presentation, as I told you, in presentation we studied that some points we studied. Look at this line. Maybe this line is clear to all now. All gibberellins are derived by the end gibberellin skeleton but synthesized by the end curine. Now you will come to know what is end curine and how gibberellin is synthesized from this end curine. Okay, so let's see in the image again. We are watching. Let's discuss again the pathway. End curine. This is the end curine, and from this end curine we are synthesizing the gibberellic acid from plastid to endoplasmic reticulum. In endoplasmic reticulum it is modified, and in the cytosol it get active. Active form is formed in the cytosol. Okay, so active form is formed in the cytosol, and in plastid the full process takes place. In endoplasmic reticulum the modification process takes place, and in the cytosol the hydroxylation process takes place, which make the process active, which make the enzyme active. I hope this is clear to all the participants. If anyone of you have any doubt, you can ask. The synthesis pathway is important. इस pathway को याद करो बच्चों. This is the most important pathway. They ask different. The 15 carbon compound is zerenyl, pharancyl, zerenyl. Then you are keep in mind that pharancyl diphosphate is a 15 carbon compound. If any one of you have any doubt, ask. You are free to ask. In the synthesis pathway, if any one of you have any doubt. Yes, please write the name of scientist. Kurosawa. Yes. I'm writing the name. Kurosawa. Kurosawa is the name of scientist. Kurosawa. Okay, no doubt. Clear to all. It is the compound. And curine is the compound. Because it enters from the plastid to the endoplasmic reticulum. It is entering from the, mainly it is the enzyme which is usually catalyze the reactions, and curine is mainly the enzyme. Okay, and it is mainly catalyzing the process that the zerenyl phosphate is entering from the plastid to the endoplasmic reticulum, and from the endoplasmic reticulum we are moving to the cytosol, and in the cytosol the process of hydroxylation takes place, and this hydroxylation makes the compound active. Students, the synthesis of gibberellin is most important. All the compounds are most important. IPP2, gibberellin synthesis, all are most important. Which compound is 10 carbon, which compound is 15 carbon, which compound is 20 carbon. Mainly these points are all, these questions are every time they ask this question. Which of the following compound is, which pathway is mainly used in the synthesis of the gibberellinic acid? Mevalonic acid pathway. Malonic and mevalonic, different, these are different pathways. Mevalonic and malonic pathway, these are different. Mevalonic is mainly used in the synthesis of the this gibberellic acid. Malonic acid pathway mainly used in the synthesis of terpenes. When we will study the secondary metabolites, at that time you will come to know shikimic acid pathway, malonic acid pathway, mevalonic acid pathway. There is difference between these pathways, mevalonic and malonic pathway. These are different. Okay, I hope synthesis is clear to all the participants. Let's move on the function. All of these are the stages. Functions we will discuss in the next class. Just take the overview. What are the functions? In next class, we will discuss the function in detail. Gibberellic acid helps in the elongation. They increase the elasticity of cell and they help in the elongation process. Okay, they helps in to break the dormancy. Seeds को अब जब जमीन में डालते हैं, when we are sowing the seeds, at that time the seeds, some of the seeds get germinated, but some seeds are not able to germinate. And to break the dormancy of seed, gibberellic acid is given. Dormancy, the inactive form of the seed. To breaking the dormancy, gibberellic acid is given to the plants. It helps in the promotion of flowering. Transport is non-polar, bidirectional. Okay, in case of oxygen, we studied that the transport is unidirectional. These are the some small points you keep in mind. In oxygen, the transport is unidirectional. On the other hand, in case of gibberellin, the transport is bidirectional in both the direction. It helps in the process of glycosylation. It helps in the germination process. In next class, we will study all the processes in detail, how they are helping in the process of germination, how they increase the elasticity of the cell wall. We will discuss these points in next class, and some important questions. We will complete the gibberellin acid. After that, we will come on the other hormone. Okay, students जो class में आते हैं, आप regularly आए class में, regularly please attend the classes. After that, you will come to know what is going on. Okay, in next class, in next class we will discuss the function of gibberellin acid. If the student joins first time, then he will not come to know what are the pathway of synthesis of gibberellic acid, and he will not come to know what is going on in the class. Okay, so be regular in classes. After that, you will come to know that in last class we studied the biosynthesis. Today we are going to study the functions of gibberellic acid. In next class we will study the cytokinin hormone. So you will come to know, if you are attending one class, in next class you are absent, then it will not create any sense. Okay, you know, you have to maintain the consistency in your classes. After that, you will come to know what is going on. Okay, sir, from my side, I have completed the topic. In next class, maybe we will finish the topic with the functions. So, students, thanks to all the students. If any one of you have any doubt, you can ask. I'm giving you 5 minutes. If any one of you have any doubt, then you can ask.
Okay, ma'am. Students, if you have any doubts regarding today's topic, you just ask ma'am in the comment section. Please comment your questions in the comment box. As ma'am has already said, please maintain the consistency in the class. Please be regular.
Students, after the consistency, you will come to know what is going on in the class. Okay, in next class, if the new student will come, he will not come to know what is synthesis of gibberellin acid, what is gibberellin acid. For him or his, the topic is new. Okay. At timings are good. Yeah. From 6 to 7, the timings are good.
Yes, ma'am. Students, if you have any doubts, please ask. Anyone? If they have any doubts, they can ask now. Or otherwise, if you have understood today's topic, you can just comment yes in the comment section. Just please respond.
Students, respond quickly.
Okay. Thank you for responding. Thank you so much. And that's all for today, guys. I hope you all enjoyed. And once again, I would like to repeat, from 10th November onwards, we are going to begin our structure class. So those who already registered, congratulations to them. And those who still not registered are willing to register, please make your registration done before 10th November. Okay. And one more thing.